Okay, so this is going to be part 3, uh, and we're going to talk about tubular reabsorption and secretion in this particular part. Okay, so in tubular reabsorption, substances must pass from filtrate in the lumen inside of the tubule across or between the tubule cells into the interstitial fluid, and finally across or in between the endothelial cells of the peritubular capillaries to re-enter into the blood. In tubular secretion, it's the opposite. Substances can be transported across tubular cells by 2 different routes. The first is paracellular. This is when substances pass between adjacent tubular cells. Tight junctions between the cells are just leaky enough to allow small ions and water to move passively between them, particularly in the proximal tubule. This requires no energy in the form of ATP, and it consists of either water moving by osmosis or solids moving within their concentration gradients. The second mechanism, or second route, is transcellular. Substances like glucose and amino acids must move through the tubule cells. So what happens is they get absorbed first through the apical membrane of the cell, the facing tubule lumen. Then it travels through the cytosol and finally exits through the basolateral membrane, the side facing the interstitial fluid. Some solids travel passively, others moving along transcellular routes travel against their concentration gradients, and these will require energy, directly or indirectly, from ATP, either through primary or secondary active transport. Secretions is active and must occur via the transcellular route across the tubule cells. Reabsorbed substances that have entered into the interstitial fluid may then cross the endothelial cells of the blood vessels and enter the blood. Substances can follow the same routes into the capillary that they followed to exit the tubule. They may take the paracellular route or the transcellular route. Generally, these processes are passive. Solids move by diffusion and water by osmosis. So here you can see the differences between the transcellular as well as the paracellular and either passive or active. Active is indicated in red here. Most substances either reabsorbed or secreted via transcellular route do so by carrier-mediated transports. These are transport proteins. In one of the following processes, you have facilitated diffusion. This is the carrier passively transports substances without the use of energy from ATP. This is typically going down concentration gradients as well. Primary active transport, this directly uses ATP to move substances against a concentration gradient. Carrier protein or pumps 2 or more substances in the same direction, which is referred to as a symport, or in opposite directions, which is referred to as an antiport. Most substances are either reabsorbed or secreted via these carrier-mediated transport, which can also include secondary active transport. This is a concentration gradient that's set up by primary active transport pump, which is used to drive the transport of a second substance against its concentration gradient. So you're not actually expelling any ATPs to move the second substance, but you are expelling ATPs to move the primary substance. Secondary active transport can't occur without primary active transport. Pumps can become saturated when all binding sites are filled. This represents their transport maximum, the maximal blood solid levels that can be transported. Substances that do not bind to the carrier protein because of saturation will most likely remain in the filtrate and eventually be eliminated from the body in urine. This is commonly seen in diabetics that have very high blood sugar levels. What happens is you are reaching your transport maximum for glucose, of bringing glucose back into the body, meaning the blood vessels, and any excess glucose that remains is just going to end up being excreted from the urine. That's one of the mechanisms in which the body can control blood glucose levels, or removing some blood glucose. So the main roles of the proximal tubule in reabsorption from filtrate back to the blood. Large quantities of ions, sodium, potassium, chloride, sulfate, and phosphate, vital to electrolyte homeostasis. Almost 100% of nutrients, including glucose, amino acids, water-soluble vitamins, and lactic acid, are reabsorbed. As I said, if you have glucose that's being excreted in your urine, that means your blood glucose levels are too high and you're exceeding the maximal levels. Bicarbonate ions are critical to the acid-base homeostasis, and about 65% of filtered water, vital to maintaining fluid homeostasis, gets reabsorbed. So sodium ion reabsorption is vital to the reabsorption of many other substances from the proximal tubule. There are two transcellular mechanisms: facilitated diffusion through ion leak channels in the tubule cell apical surface, as well as active transport via 3 carrier proteins on the apical surfaces for sodium ions. The 3 carrier proteins are carrier proteins specific for sodium ions. This will enable facilitated diffusion of sodium ions from the filtrate into the tubule cells. You then have sodium symporters. These transport sodium ions from the filtrate into the tubule cell with other substances like glucose. And then you have the sodium hydrogen antiporters. These bring sodium ions into the cell from the filtrate, as well as secrete hydrogen ions into the filtrate. All 3 carrier proteins transport sodium ions down their concentration gradients into the tubule cells. You then have the sodium-potassium pump. This creates and maintains a gradient by transporting sodium ions into the interstitial fluid through the basolateral membrane. The pumps create relatively low sodium ion concentrations in the proximal tubule cells. Sodium ion concentration in the filtrate is higher. The gradient is critical for secondary active transport from many other solids. Reabsorption of organic solids and ions will occur in the first half of the proximal tubule. Sodium glucose symporters use sodium ion gradient to move both sodium ions as well as glucose molecules by secondary active transport. Amino acids, lactic acid, sulfate ions, and phosphate ions are transported by secondary active transport as well. The reabsorption of sodium ions also leads to the reabsorption of anions, such as chloride ions. As sodium ions are passively transported out of the tubule lumen, the lumen accumulates a net negative charge. This creates an electrical gradient that pushes negatively charged chloride ions across the epithelium through the paracellular routes. The chloride ions also follow sodium ions into the interstitial fluid and the plasma. Okay, so here you can see the reabsorption and secretion within the proximal tubule. Bicarbonate ion reabsorption. This occurs as a result of the sodium and hydrogen antiporter activity and reversible chemical reaction that we saw with the transport of CO2 with the blood vessels. Hydrogen ions are secreted into the filtrate combined with bicarbonate ions forming carbonic acid. The carbonic acid is converted to CO2 and water by an enzyme referred to as carbonic anhydrase on the apical tubular cell membrane. Carbon dioxide diffuses into the cytosol, where it is again combined with water to form bicarbonate and hydrogen ions, catalyzed by carbonic anhydrase. This process repeats as hydrogen ions are again secreted back into the tubular lumen. This allows the cells of the proximal tubule to reabsorb about 90% of bicarbonate ions. This reabsorption is a key component of the body's ability to maintain pH of blood within a very defined range of 7.35 to 7.45. And here you can see bicarbonate reabsorption in the proximal tubule. Obligatory water reabsorption and the effect on electrolytes. This occurs in the second half of the proximal tubule, where sodium ions, glucose, and other organic molecules have already been reabsorbed. Water is obliged to follow reabsorbed substances. Concentration gradient is established that favors passive movement of water out of the filtrate by osmosis through paracellular and transcellular routes. Aquaporins, which are water channels in both the apical and basolateral cell membranes, greatly increase the reabsorption of water. As water is reabsorbed, concentration of magnesium, calcium, and potassium ions increases in the filtrate. And this actually will favor reabsorption of these ions by diffusion into or between the tubule cells. And you can see the water movement here based on other things moving through. Now, secretion in the proximal tubule includes hydrogen ions, which was discussed a few minutes ago, many drugs, and various nitrogenous waste. In the first half of the proximal tubule, most uric acid infiltrate is reabsorbed. Nearly all of it is secreted back into the filtrate in the second half of the tubule. Ammonia ions, creatinine, and small amounts of urea are also secreted. Drugs such as penicillin and morphine have significant renal secretion and must be taken often, typically 3 to 5 times per day, because it gets lost through the secretion and must be replaced in order to maintain the relatively constant blood levels. So now let's talk about reabsorption in the nephron loop. Once filtrate reaches the nephron loop, 60 to 70% of water and electrolytes must be reabsorbed and returned to the blood. About 20% of water and 25% of sodium and chloride ions are reabsorbed from the loop. In the proximal tubule, water reabsorption is proportional to solid reabsorption. So filtrate has the same osmolarity as the interstitial fluid, which is about 300 milliosmoles. In a nephron loop, however, filtrate's osmolarity changes. This is due to differing permeabilities in the ascending and descending limbs. The thin descending limb of the nephron loop is freely permeable to water, but much less permeable to solids such as sodium and chloride ions. Water can move out of the thin descending limb cells by osmosis. A few solids will follow. This causes the osmolarity of the filtrate to increase as it passes down the descending and round bends of the loop. Cells of the thick ascending limb are impermeable to water. They transport sodium chloride into the tubule cells with the sodium, potassium, and chloride symporters. This brings 1 sodium, 1 potassium, and 2 chloride ions into the tubule cell. This relies on the sodium-potassium pump on the basolateral membrane to create favorable sodium ion concentrations. The sodium-potassium pumps drive potassium ions back into the tubule cell, so there is not much net reabsorption of the potassium ions. As the filtrate passes through the ascending limb, it loses solids and gradually becomes less concentrated as the ions are pumped into the interstitial fluid. By the time filtrate reaches a segment of distal tubule, about 85% of water and 90% of sodium ions have been reabsorbed. The rate of filtrate flow in this part of the tubule is significantly slower, about 20 mils per minute, than it was in the earlier proximal tubule, which was about 120 mils per minute. The early distal tube is structurally and functionally similar to that of the ascending limb of the nephron. The latter portion of the distal tubule is very similar to the cortical collecting duct. So we're going to discuss that together here. The medullary collecting system differs structurally and functionally from these two areas. This is for the juxtamedullary nephrons. So principal cells of the late distal tubule and collecting duct have a hormone receptor that determines their function. Majority of their activity is regulated by hormones in order to fine-tune water, electrolyte, and acid-base balance. Water absorption is therefore facultative, based on the body's needs. You have aldosterone. Remember I said earlier that when you get to this point, reabsorption is going to be hormonally controlled. So aldosterone, this is a steroid hormone that is made by and releases from the adrenal cortex. It increases the permeability of sodium ions and the number of sodium-potassium pumps, increases the reabsorption of sodium ions and the secretion of potassium ions. As sodium ions are reabsorbed, water and chloride ions passively follow, especially if a antidiuretic hormone or ADH is present. This stimulates the secretion of the hydrogen ions into the filtrate by the intercalated cells. ADH is made by hypothalamus and it's secreted by posterior pituitary. This causes water reabsorption, reduces urine output, exerts these effects by causing aquaporins to be inserted into the apical membranes of the principal cells. In the absence of ADH, the principal cells are barely permeable to water, and larger volume of water is actually lost in urine. ANP will stimulate urinary excretion of sodium ions. This inhibits the release of both aldosterone and ADH and causes fewer sodium ions and also less water to be reabsorbed. This results in more water and sodium excretion. Now, the medullary collecting system, these are those juxtamedullary nephrons. This is the last chance for the regulation of fluid, electrolyte, and acid-base balance before the filtrate becomes becomes urine. The principal cells in this part of the system are impermeable to water in the absence of ADH. They are permeable to urea, which allows urea to be reabsorbed passively into the interstitial fluid, and they continue to reabsorb ions such as sodium chloride and bicarbonate from filtrate. Both principal and intercalated cells are actively going to secrete hydrogen ions into the filtrate against the steep concentration gradient. It can actually increase the proton concentration in the filtrate by as much as 900 times. Cells of the proximal tubule will secrete ions as mechanisms for reabsorbing bicarbonate ions, while cells of the distal tubule will actively secrete hydrogen ions. So when your blood pH decreases, it becomes too acidic, enzymes in the tubule cells will remove the amino group from amino acids glutamine. This generates 2 ammonia and 2 bicarbonate molecules. Ammonia is secreted into the filtrate and the bicarbonate is reabsorbed into the blood. When blood pH, ammonia in the filtrate acts as a buffer that binds to the hydrogen ions, forming ammonium ion. Ammonia, buffering, and bicarbonate reabsorption together bring pH back up towards a normal range. If the blood pH increases, becoming too alkaline, the tubule cells will reabsorb less bicarbonate ions from filtrate and lowers the blood pH as these ions are excreted in the urine. So here, I know we talked about a lot of things moving here. So this is actually a very good figure. And it really summarizes everything that is moving either from the filtrate to the blood vessel or from the blood vessel back into the filtrate, okay? whether it's water, sodium, glucose, various other ions. And it also will tell you if it's by hormonal regulation or not. It's going to be by hormonal regulation in that collecting duct area. Okay, so this really summarizes all of tubular reabsorption and secretion. So once you sort of have gone through these slides and listened to this recording and then have read through this, this would be a good slide to really just have what's moving when and where. The green arrows mean it's moving passively, usually down concentration gradients or facilitated diffusion. The red arrows mean it's either primary or secondary active transports. And that'll be the end of recording number 3.